I would like to thank the organizers for the opportunity for me to present on behalf of the project team on our theme-based research scheme project on diabetes, heart, and kidney complications. But before I start, I would like to describe the motivation of us doing this work. As clinicians looking after people with diabetes, we are moved and touched by the plight and need of our patients with diabetes, such as Mr. WK, who is a gentleman I saw a few years back. He was 36 years old at the time, who has a strong family history of diabetes, and he was diagnosed with type 2 diabetes at the age of 27. Unfortunately, he had poor glucose control, and by the time I saw him, he already had eye complications, kidney complications, high blood pressure, and hypercholesterolemia. And his kidney biopsy confirmed the damage to his kidneys were due to his diabetes. But despite these complications, he was not motivated to improve his glucose control, and he had poor glucose as well as cholesterol control, and his renal function suggests that he's already reaching end-stage renal disease. And even though it's our first encounter, I already know that within a few years' time, he would be entering dialysis and would have to spend the rest of his life next to a dialysis machine. Not good prospect for a young gentleman. That begs the question that had he known and had his doctors know this is what his outcome might be, could things have been different? What could we do to improve his outcome? So it's against this background that I would like to describe the burden of diabetes and its complications, and also the main aims of our TRS project. I will give an overview of some of the major research findings and achievements of the project and some of the related output, and how we're translating these to try to improve care for patients with diabetes, such as the one I described. The WHO has estimated that there are currently 422 million people with diabetes globally, affecting approximately 1 in 11. The great majority, over 95% of these people, have type 2 diabetes. If we look at the global burden of diabetes, we can see that the largest number of people with diabetes living in the world reside in China, India, and the United States. In China alone, you can appreciate that over the last 3 decades, there has been a marked increase in the prevalence of diabetes, which currently affects approximately 10.9% of the population. That translates to over 110 million people with diabetes in China alone, and more than 380 million with prediabetes. Diabetes in Asian individuals are characterized by a young age of onset, in addition to high rates of kidney complications. Due to the chronic high glucose seen in diabetes, diabetes can damage different organs, including the eye, including kidneys, and diabetic kidney disease is now accounting for about half of the people entering dialysis program in the Asia region. In addition, diabetes can damage the nerves and leading to debilitating lower limb amputation, and diabetes increases the risk of heart disease and stroke by approximately 2 to 4-fold. In addition, the high glucose also increases the risk of infection, dementia, osteoporosis, periodontitis, atrial fibrillation, and cancer. And it's safe to say that no organ from the body is really protected from the damaging effects of high glucose. In Hong Kong, diabetes accounts for approximately 200,000 hospital admissions per year, with an estimated healthcare burden of $5 billion per year. The great majority of this is due to the management of diabetes complication, in particular the heart and kidney complication. Amongst people presenting with heart and kidney problems to the hospital authority, a large majority of these are due to diabetes. And in fact, heart and kidney complications alone account for 50% of deaths in people with diabetes. It is therefore our aims in our project to try to define molecular symmetry of diabetic cardiovascular and kidney complication to identify some of the key pathogenesis pathways. In addition, we want to establish a territory-wide registry and biobank for replicating our biomarker discovered. We also want to build on previous work on examining SMED3 and PPAR delta as candidate pathways for development of diabetic complications. Most importantly, we want to explore how we can try to translate some of these findings to improve prediction and treatment, with the ultimate aim to reduce the burden of diabetic kidney disease and cardiovascular disease in diabetes. The strategy we've taken is to leverage on a unique resource, the Hong Kong Diabetes Registry, which was initiated by co-PI Professor Juliana Chan back in 1995, where since then we have established a cohort of more than 10,000 patients with diabetes with prospective follow-up, consensus samples for biobanking and genetic studies, and also detailed clinical information and prospective follow-up. During the project, we applied multi-omics approach to interrogate the genome, the methylome, microRNA, and transcriptome, and also to integrate these findings and working together with animal models of diabetes to understand functional pathways of both candidate pathways, such as SMED3 and PPAR delta, as well as novel discovered pathways from the project. To replicate these findings, we also work with investigators across different hospitals in Hong Kong in order to build a replication cohort Hong Kong Diabetes Biobank, as well as undertaking replication with our external collaborators internationally. The project is led by a steering committee of the co-PIs, advised by international advisors, and grouped into specific working groups according to the area of expertise. So for example, in work led by Professor Huang Yu in one of the basic animal studies examining the role of PPAR delta, we were able to expand on the previous work from Professor Huang Yu in trying to understand the role of PPAR delta endothelial dysfunction in diabetes. And Professor Huang Yu found that PPAR delta, in fact, is a key mediator for mediating the beneficial effects of exercise in actually damping down ER stress and actually reducing oxidative stress, as well as endothelial dysfunction in relation to metabolic diseases. Another example, Professor Lan's group, who has pioneered work identifying SMED3 as a downstream target of the fibrogenic cytokine TGF-beta and identifying downstream genes from SMED3, such as long non-coding RNA, which are related to renal fibrosis, were able to take this work forward and examine the role of SMED3 in a type 2 diabetes animal model. In this work, Professor Lan crossed the DBDB mouse, which is an animal model of type 2 diabetes, with a SMED3 knockout background in order to generate SMED3 knockout mouse on a diabetes background. These mice were examined for development of kidney and heart complication, as well as molecular changes in different tissues in these animals. Interestingly, the diabetes mouse, as shown here, the DBDB mouse compared with the wild type SMED3 wild type mouse, have an obese and metabolic adverse phenotype with hyperglycemia, obesity, as well as dyslipidemia. But interestingly, crossing this into a SMED3 knockout background substantially improved the metabolic phenotype of these mice, which were no longer overweight, and in fact, shown in blue, had complete normalization of glucose despite being from a DBDB background. Needless to say, these animals were protected from the development of kidney and heart complication, but most excitingly, led to this new avenue of research into why SMED3 is involved in glucose homeostasis. In subsequent work, Professor Lan's team have been able to identify the PAX6 gene, an important transcription factor involved in beta cell proliferation, as the downstream target of SMED3. And as you can see here, in wild-type DBDB mouse, there is substantial loss of beta cell as well as insulin production, but this is substantially improved in SMED3 knockout mouse on the DBDB background with normal production of insulin as well as improved expression of PAX6. This has now led to downstream work to not only explore the role of SMED3 in prevention of diabetes complication, but also in metabolic diseases. Going back to the human studies, our aims were to identify individuals at risk of diabetic kidney disease, and in one of the early work, trying to look at a cohort of patients free of diabetic kidney disease using analysis approaches such as latent trajectory, we were able to characterize that the great majority of individuals, in fact, have fairly gradual decline in renal function. But we were able to identify a subgroup shown here who have accelerated decline in renal function and therefore progress to kidney disease and end-stage renal disease rather rapidly, like the patient I described. In addition, there were some intermediate phenotypes. And having identified these different patterns of renal disease progression, we were able to identify the substantial heterogeneity in risk of kidney disease, as well as the prognosis of these people with more rapid renal decline who have higher risk of dying and also other complications. We were also able to identify genetic risk factors that are associated with this rapid decline phenotype. Furthermore, utilizing our initial findings, we were able to generate genetic risk score, which can help us stratify individuals for their subsequent risk of kidney disease. So shown here, for example, individuals with the highest tertile of the genetic risk score based on 24 SNPs, they have approximately 5-fold higher risk of progressing to end-stage renal disease during follow-up compared with individuals with the lowest risk. And this is after adjustment for duration of diabetes and other risk factors such as smoking, glucose control, and baseline renal function, showing that these risk factors can help us stratify patients in clinical practice. Furthermore, in order to further undertake functional studies to understand the functional significance of some of these genomic regions identified, in collaboration with clinician Professor Sito, we've been able to undertake transcriptome analysis using laser capture microdissection of renal biopsy samples and identify the gene changes associated with diabetic kidney disease in both renal tubule and glomeruli, and to separate some of these gene changes from non-diabetic damage, such as those caused by hypertension. And this will help us to identify key pathways involved in the pathogenesis of diabetic kidney disease by integrating with the genomic data. In addition, leveraging on the large amount of whole genome genotyping data generated, we were also able to advance our understanding of type 2 diabetes. So in this multi-institution international collaboration, we undertook a meta-analysis of type 2 diabetes, including more than 77,000 individuals with type 2 diabetes and more than 350,000 control, leading to the identification of 300 signals at 183 loci, of which 61 were novel, giving lots of new hits and in terms of findings in terms of candidate genes associated with type 2 diabetes, and also giving us a comprehensive picture of some of the differences in genetic architecture in type 2 diabetes in Asians compared with in European individuals. The project team also were able to leverage on the large number of genotyping data generated to also undertake method development. So for example, in work led by Professor Wei-Chuan Yu and also his student Sang Chang at the Hong Kong UST, they were able to develop methods to examine region-based interaction detection in genome-wide case control study. And this method will enable investigators to examine gene-gene interaction using similar data sets from genome-wide association studies. Trying to replicate key biomarkers discovered in the project is one of the major aims of the project. And to do this, we need to have independent cohorts. So in collaboration with colleagues from 11 diabetes centers and 3 renal units, the project team during the project period was able to initiate the Hong Kong Diabetes Biobank, which recruited more than 22,000 subjects with similar collection of information like the Hong Kong Diabetes Register, with prospective follow-up of biobank samples, with standardized procedures for collection, detailed phenotype information, electronic health record linkage, and also we were able to develop a pipeline for high throughput DNA extraction as the samples come in order to generate the DNA to put them through to whole genome genotyping for replication studies. This has benefited the project immensely, as shown in this example, where we've been able to use samples from the Hong Kong Diabetes Biobank during the project period to conduct replication study. So in a meta-analysis of coronary artery disease risk in type 2 diabetes, in our original discovery from Hong Kong Diabetes Registry, we had 6,000 patients genotype, of which around 1,000 had type 2 diabetes with coronary artery disease and 3,600 free of coronary artery disease. And using data generated from the Hong Kong Diabetes Biobank, we were able to genotype another 7,000 of the first lot of patients and integrate the genotyping data into a meta-analysis. In addition to looking at heart and kidney complication, we also took the opportunity to look at some of the other phenotypes from our rich data set to identify genetic factors associated with other outcomes in patients with diabetes. So for example, patients with diabetes often experience drug failure and need to go onto insulin. And using data from the register as well as the newly established biobank, we were able to interrogate the data set to establish some of the genetic factors associated with drug failure as well as need for insulin. So for example, using genetic factors associated with diabetes risk or SNPs associated with metformin response, we were able to construct genetic risk score that would allow us to predict failure of oral treatment in these patients who would need to go on insulin sooner, whereby individuals at the highest risk have approximately 30 to 40% higher risk of progressing to need for insulin use. And these can be used in clinical practice to improve clinical treatment and reduce clinical inertia. So these are some examples where findings from the study from the project has enabled us to develop either biomarkers or tests that will help us stratify patients in terms of risk and also enable us to have better subphenotyping for better management with our ultimate aim to deliver precision medicine in diabetes. As overall summary of the project, during the project period up to December 2019, the project has delivered 66 publication and 3 book chapters with 48 abstract presentations, and also has supported the training of 30 postgraduate students across the different institutions, with 24 awards received by the students, 5 software packages, as well as more than 100 major invited talks at international conferences, with more that has followed subsequently. The project has also, several members of the project team has also been received recognition. For example, our co-PI, Professor Juliana Chan, was recognized by the American Diabetes Association with the 2019 Harold Rifkin Award, one of their most distinguished awards from the association, as well as the Peter Bennett Award, and also from Monash University for her contribution to diabetes care and research. In addition, Professor Wang Yu has also received several awards, including the Higher Education Outstanding Scientific Research Output Award from the Ministry of Education of People's Republic of China. I've also received recognition from the Hong Kong College of Physicians, as well as the Croucher Foundation Senior Medical Research Fellowship. The project team, by working on different aspects, including epigenomics of diabetes, was able to link up our different colleagues and investigators internationally who are interested in this area, and was able to initiate a new Golden Research Conference on epigenomics of diabetes and related metabolic trait, with the first meeting hosted in Hong Kong in May 2018. And the project team also benefited from the guidance of our international advisors, and met every year through our annual Diabetes and Cardiovascular Risk Factor East Meets West Conference. And this is a meeting of the steering committee and the co-PIs with the international advisors in 2016. We've also embarked on collaboration with AstraZeneca to undertake whole genome sequencing study for diabetic kidney disease, and with the aim to functionally characterize 1 to 2 novel drug targets for developing treatment for diabetic kidney disease. The project team has also been engaged in major global diabetes initiative. So Professor Juliana Chan has been invited by the Lancet to lead the Lancet Commission on Diabetes, with the report Using Data to Transform Diabetes and Patient Lives, recently published on 13th of November 2020, with several members of the TRS team also involved in this initiative on the Lancet Commission. And this is now in discussion with other partners such as the WHO for implementing some of the recommendations to improve diabetes treatment in different regions of the world. There's also increasing focus on the need for precision medicine in diabetes, and I've been fortunate to be invited by the American Diabetes Association to lead one of the subgroups on diabetes complication in the upcoming consensus report on precision medicine in diabetes. But taking our findings from our project has led to the development of a customized Asian screening array that we have put together, including some of the findings from our genomic studies, which we have used to genotype the remaining samples from the Hong Kong Diabetes Biobank, as well as clinical use. And this work is now being taken forward with support from the RGC Research Impact Fund. An example of how we're applying this is in this work commissioned by the Food and Health Bureau and led by Professor Juliana Chan, where we apply whole genome sequencing to identify novel genes for young onset diabetes, as well as conducting a randomized clinical trial incorporating some of these genetic risk score, as well as the customized genotyping array to deliver genetic marker to improve care for patients such as Mr. WK. The project team is continuing to engage with stakeholders both locally and internationally to try to implement some of the findings as well as approaches identified in delivering precision medicine in diabetes. This would not have been possible without the tireless work from all the members from the project team, and I would like to take this opportunity to, in particular, thank all the team members with all the co-PIs and also the steering committee, as well as all our young investigators and also our students who have contributed to the different aspects of the work during the project period. We've also been able to establish different collaborations from international institutions, as well as large genetic consortia, to try to advance the understanding of genetics of diabetes, as well as different diabetes complications and outcomes. And hopefully with these, we can continue to improve and apply some of these knowledge to improve the care of patients with diabetes. I'd like to finish by acknowledging again many team members who have contributed to this work. In particular, members from different hospitals across Hong Kong who have contributed to the Hong Kong Diabetes Biobank, as well as members of the theme-based project team, and of course, the support from the theme-based research scheme, which has made all this possible. And with that, I would like to finish and thank you for your kind attention. And I'll be happy to take any questions.
I would like to ask you maybe start with, this is obviously, you've been working on this for the last 4 to 5 years. Can you just share with us the challenges in actually integrating this very multidimensional knowledge, all the way from basic science, data science, and also clinical implementation? And how do you see such integrative work can really transform care?
Thank you, Professor Chan, for this important question. I think that is one of the biggest challenge that many of us working in this sort of area are facing. Much of what I have actually presented, given the large amount of data generated, remains insights that have been generated based on one level of information or two levels of information, i.e. like just the genome, or the genome with the transcriptome, or the genome with the methylome. Integrating really all the dimensions of the different layers of information really is where the challenge is. And I think currently there are still only limited methodologies to do this. So currently many of the methods that we're using are focusing still on 2 or 3 layers and not really fully integrating all the data to maximize insight. But I think we are all working on methods to try to integrate these so that on an individual level, the layering of all this information can help advance some of the insights and care. And certainly now by looking at individual level, how all these layers of information are relevant is what we're currently working on. And I think new approaches and methods that would facilitate this kind of analysis would be quite important.
Obviously, these are very, very resource demanding work, not just in terms of time, in terms of thinking, in terms of different people, but it is really, very encouraging and indeed rewarding to see, how many students and fellows, who have been sort of trained through this very complex projects, and how discoveries have been made in terms of potential biomarkers, potential even drug targets, for example, SMAP3, and in terms of actually also understanding disease mechanisms. But, in the immediate sort of next 3 to 5 years, do you see opportunity that some of these discoveries can be translated to clinical care? And as such, how can we improve, or the government or the industry can improve this environment so that they can really be put into practice while we wait for new drug discovery, for example?
That's a very important question. Thank you. So I think for many common disease like diabetes, precision medicine is just something that is gathering momentum. So we're very much behind like the cancer field in terms of appreciating the importance of molecular signatures and also subphenotype and how that will guide treatment. But I think there are exciting opportunities ahead. I think, for example, we need 1 or 2 examples of where knowledge of the molecular basis and the genetic basis and genetic risk score can actually impact on outcome. And I think hopefully the trial done by ourselves and others will generate some of this evidence of how biomarkers can actually alter care. We also need some form of consensus as to what are the biomarkers that would help improve clinical care. And I think to that end, it's exciting that the American Diabetes Association and the European Association of Study of Diabetes, so one of the 2 large academic bodies, have now put together this initiative that includes more than 200 investigators across the world in generating, hopefully, this consensus in what are some of the biomarkers, in reviewing the literature, having some consensus, what are the biomarkers that would improve care in diabetes. And being involved in this initiative, hopefully, we will have a better idea of how to integrate biomarkers into improving care, and that will help disseminate this practice as well as application in improving care in diabetes. So I think those are some of the short-term, hopefully short-term outcomes that some of the findings from the study that we're undertaking would also be able to link into clinical practice to change practice. But I think certainly genomic medicine is here to stay. And I think, for example, the hospital authority also is reconsidering some of the areas of genomic medicine that can be incorporated. And many of us are involved in different aspects in terms of training of future physicians, in terms of their understanding of genomic medicine, and as well as also in trying to find areas where these sort of practice can translate into clinical care.
I think this is exactly the next sort of the next challenge, and that is, we now have so much knowledge, and given that, we have like 10% of people with diabetes, and that, 30% of them are at risk, and that's among those people who have diabetes, we always want to find those who are really at risk for complications, etc. We clearly need more people to deliver, right, these technologies, even if they are developed, right? We need people to articulate and to also, sort of, use it to actually improve care. And I think this is really where, the UGC and also I think the university as well as, needs to really prepare our students, not just the medical students, but really all the healthcare disciplines to understand, how these knowledge are important, right? How can we actually use it? And so I think this is, and of course, at the same time, how do you see that we can educate the public, for example, because but I think now, for example, with COVID-19, the public are becoming more aware of what's like phase 2, phase 3 studies, etc. Do you see the public will be able to learn more about how these genetic markers can, affect their risk and how they can do something to manage it?
Certainly, I think this also relates to a question that just has been posted of how we educate the public in terms of risk. I think they need to have an understanding of risk of disease, what diabetes, how diabetes may impact them, and also do all the necessary clinical screening. I think when the biomarkers become validated and are considered valid for clinical use, then I think these hopefully will be integrated into clinical care pathways. And then the public will benefit from that. Specifically with regard to the question about, I think the question is on whether people with family history of diabetes should have nutrigenomic analysis. I think currently the data supporting that is not strong in terms of the benefit of genetic factors in altering dietary responses, though this is an area of active research. And there certainly are some exciting findings from some of the algorithms that are being developed to stratify people's response to diet. But I think, again, this has some way to go to have some consensus as to what are the biomarkers that really would impact and improve care.